Haplotype. What can a haplotype tell us? Firstly, since haplotypes are evolutionarily conserved, they can reveal the trajectory of ancestor migration. Secondly, the same haplotype is very rarely generated in different regions. Therefore, it can clarify racial or ethnic differences. Lastly, haplotypes can define individual differences by the diversity generated this way. What is a haplotype? A haplotype refers to a haploid genotype. Since we have two copies of each chromosome, we also have two copies of every gene in most cells. As you can see, autosomal DNA has been inherited from the ancestors of both parents. On the other hand, while chromosomal DNA and mitochondrial DNA have been inherited from male and female lineages, respectively. A haplotype is a set of closely related genetic markers on one chromosome that tend to be inherited together without recombination in the flow of such inheritance patterns. Haplotype also refers to a set of DNA variants along a single chromosome. As you know, genetic information is encoded in DNA by a sequence of 4 bases: adenine, thymine, guanine, and cytosine, and there are individual differences in this sequence. The differences in the genetic code that occur at the frequency of 1% or more of the population are defined as single nucleotide polymorphisms. They occur roughly every 500 to 1,000 base pairs and are thought to be a factor that causes individual differences. As the figure shows, a haplotype refers to a physical grouping of genetic variants such as SNPs, and it can be limited to a single gene, or it can be larger and include multiple genes on the same segment of chromosome. Next, as an example of a haplotype, we will describe HLA, a human leukocyte antigen. Here is an example of the HLA haplotypes of one parent and their children. Circles, squares, triangles, and stars indicate different HLA haplotypes. Now, consider the HLA haplotype using mother as an example. As you can see, HLA complex is located on the short arm of chromosome 6. Class 1 regions include the classical HLA-A, HLA-B, and HLA-C loci, and Class 2 regions include the HLA-DP, HLA-DQ, and HLA-DR loci. And HLA-RL is defined by the combination of non-synonymous SNPs within the coding regions of HLA genes. In this definition, HLA-RL corresponds to a unique HLA protein. HLA haplotype is defined by the combination of HLA-RLs in HLA region. This is a graph showing the number of HLA-RLs discovered so far by year. The number of HLA-RLs discovered has been increasing with techniques such as next-generation sequencing. Increased variation at the HLA-RL level implies increased variation at the HLA haplotype level. As a result, it is believed that there are tens of thousands of HLA haplotypes. Considering such diversity of HLA haplotypes is very important in medical practice. Diversity in HLA haplotypes causes individual differences in immune responses. Therefore, the HLA haplotype is useful in tissue and cell matching, in which tissues and cells from one individual are analyzed to determine whether they can be successfully transplanted to another individual. In addition, differences in HLA haplotypes cause differences in people in responsiveness to the vaccine. Furthermore, since a lot of HLA genes are associated with human diseases, HLA haplotypes can be used as a disease risk marker and is expected to be useful for disease prevention. Thank you for watching this video.